In this section, I'm going to talk about bacterial replication. Binary fission is kind of like the mitosis of bacterial growth. So binary fission is the process of cell division in bacteria. Here you can see a cell. The cell is going to elongate, and then the genetic material is going to be replicated. The genetic material is, the copied chromosomes are going to separate toward opposite ends of the cell, and then a band is going to form in the center of the cell. Eventually that band will pinch in and shrink, and those cells will divide in half. The two daughter cells will then be pulled apart and free to be divided and to do their normal cell function. The generation time or doubling time is the time required for a complete fission cycle. So binary fission, we call it a fission cycle. So it's the time to double. So the time it takes for one cell to turn into two cells. Each new fission cycle doubles the population. As long as the environment remains favorable, the doubling effect can continue at a constant rate. So here you can see the number of cells. After 5 generations, you're already at 32 cells, and you can see how this can grow exponentially. Doubling time or generation time of E. coli is anywhere between 10 to 20 minutes. And then different organisms are going to have different generation times. Mycobacterium tuberculosis has a really long generation time, so the time to double is really long. Mycobacterium leprae, that causes leprosy or Hansen's disease, has a 14-day doubling time. So you can see that those ones that have very short doubling times can turn into a lot of cells very quickly, while the ones who have very long doubling times are going to replicate at a very slow rate. Exponential growth is the growth pattern of microbes. It's useful to express the populations of microbes as exponents or logarithms. So if we look at time and we look at this exponential growth, it takes some time before growth can happen, but once it starts, it's going to rapidly divide and you're going to see numbers skyrocket very quickly. The growth curve is a predictable pattern of growth in a population of bacteria. So it's basically a diagram of the number of cells and how they're expanding, how they're increasing. The viable cell count technique is traditionally used to observe population growth pattern. The total number of cells is counted over a given time period, and this is a fundamental method of laboratory microbiology. So here you can see if you have this inoculated flask, you put it into new media. This will not have any growth at 60 minutes, but then if you let it grow for 120 minutes and incubate it, you can see a couple cells there that have turned into colonies. If you let it sit for 180 minutes and then you plate it, you can see more. After 240 minutes, you can see even more, and you can kind of go up from there to eventually not being able to count how many colonies are there. So this is a way to express a growth curve. The common growth curve has different phases. So you have the lag, log, stationary, death, and then the after phase. So some are going to remain viable after the fact. During the lag phase, this is a flat period on the graph when the population appears to not be growing. If you just inoculate bacteria into a new medium, they need time to adjust to their new environment. So throwing bacteria into fresh, brand new, sterile media, they're not going to start dividing immediately. They need to sense that there's nutrients around each other, and they're going to evaluate their environment. So they don't replicate, and you're not going to see a lot of growth. After a period of time, you're going to see exponential growth, and this is the log phase. So you're going to see that they're completely adjusted, and now they're going to start dividing. So you're going to see bacterial numbers increase. Eventually, they're going to realize that the nutrients in the media are limited. So there's only so much glucose, or there's only so much protein inside of that media tube. You can only fit so much in here. So when those nutrients start to become limited, some of the bacterial cells are going to start to die off, while others are going to still reproduce. So you're going to have a leveling off of the growth curve. And then you have the death phase. So limiting factors intensify and the cells begin to die at an exponential rate. So all the nutrients are used up now and they're just going to start to be rapid death. The curve is going to dip downward. After that, there's still some bacteria that will be able to mutate to be able to still eat the dead or still survive in the absence of the original nutrient. They'll find something else in the media to live on, but eventually everything in the tube will die. The importance of the growth curve: antimicrobial agents rapidly accelerate the death phase. Growth patterns can correspond with the stages of infection as well. So with the exception of this viable growth curve, this viable plate count that we talked about where you incubate at certain periods of time, there are different ways to enumerate bacterial numbers. Turbatometry is talking about taking a tube of clear nutrient solution and investigating the light that passes through it and comparing that to a cloudy solution. And it becomes cloudy when bacteria are present in the solution. The greater the turbidity, the larger the population size. So here you can see turbidity. You use a spectrophotometer. The spectrophotometer will pass light through, and then it's going to record or process the percent of light that's transmitted. If it is clear, light's going to go straight through it. If it's cloudy, light's going to take a lot longer to get through, and not a lot of light is going to make it through. So we can use the measurements from turbidity in order to determine concentrations. The problem with this is this still counts living and dead cells. So it's cloudy. We don't know if it's cloudy because the cells are alive or because the cells are dead. You can also perform a direct or total cell count, and you can count cells in a sample microscopically. This is a picture of what that looks like. So you're going to plate a known concentration or put a known concentration of cells on here, and then you're going to count. So you put 10 microliters, for example, on this counting plate, and then you're going to start to count bacterial cells. And then from the known amount that you've plated, you can do calculations to determine what the total count was in the tube. Now again, you're counting both living and dead cells. Living and dead cells with bacteria look identical. So you can't tell the difference between a living and dead cell just by looking at it. Now, if this were a human cell, living cells usually absorb or do not absorb stain, while dead cells absorb stain. So you can count living versus dead human cells by looking at whether they have stain inside of them or not. Because when they die, their cell membranes become leaky and then they're prone to suck up the stain in the background. Bacterial cells don't do that. So whether they are living or dead, they're going to look the same. We can count them using a flow cytometer. This is also a method of counting white blood cells and erythrocytes. So this is just a common cell counter. We can label them with certain antibodies, which are going to fluoresce certain colors or look for certain markers and so forth, and we can count using a flow cytometer. But again, this is going to count both living and dead cells. The only way to count just the living cells is by performing a viable cell count. Viable cell count is a little different from the viable plate count that we just talked about. So viable plate count is used for determining the growth of the microorganism. A viable cell count, which I'll abbreviate a VCC, viable cell count, is when we take an original culture and then we serial dilute it. So we're going to dilute it. We take 1 mil from this to this, mix it up, then we take 1 mil from here to here, and we kind of piggyback them, and we dilute it down to a known concentration. And then we're going to plate these diluents on various plates. And when they're plated, we can perform some calculations. So we can calculate, based on the number of colonies that end up growing from this plate, the number of living cells that were in the tube, the original culture tube. How do we know that they're living? Well, we're counting what grew on the plate. If they're dead cells in here, they're not going to grow on the plate. So this allows us to only count living cells on the plate. We will be performing this procedure in lab. New methods for analyzing population growth include polymerase chain reaction. We can do a quantitative polymerase chain reaction that can quantify bacteria and other microorganisms in the environment. without isolating them or culturing them. We look for their pieces of DNA and we count how many pieces of DNA are produced, and then we can use certain calculations to determine their concentration in the environment. We can also test for ATP. So this is used in food and pharmaceutical industries and may be used for rapid quantification of microbes in other environmental samples. But we have developed tests that measure ATP concentrations.